Hello and very warm welcome to all the participants over there. Today we are going to discuss on one of the most important topic which calls as research enhancement. So if you guys are the upcoming researcher and you guys are really focusing to make certain kind of project work or else to lead yourself with the novel findings in the research and move ahead for the publication quality work, so in that context, there is one of the most important step to carry out is nothing but the um, literature survey. So in that context, today we are going to discuss on one of the most important and relevant topic, that how we can enhance our research using the bioinformatics tools, software, and even the different approaches which are available in the current era. So welcome back all of you guys. Myself Dr. Nilofer. I'm a bioinformatics scientist at Biotecnika. Today we are going to discuss on the topic, uh, as I mentioned, that is a research enhancement discussion. And in that context, today we have taken one of the reference paper where the major of the work is done and done the kind of the novel findings using the in vitro techniques and the tools. Then we are going to see that if these are nothing but the in vitro methods or the techniques available, then how we can take help of the bioinformatics approach to enhance this research with the more accuracy, and even the time consuming process can be overcome, right? So as you all can see that the paper is get published in International Journal of Food Properties, and the paper entitled with a phytochemical analysis, antioxidant, antimicrobial, and toxicity study. Just have a look, guys, here whatever that's been discussed. It's covering about like the phytochemical study, and they are including this kind of phytochemical study with respect to the antioxidant and even the antimicrobial study with the toxicity kind of approaches. So these are the things which they have take care um, with the data sets accordingly to perform the phytochemical analysis for the research. So basically the plant which they have used is a schema volichi, which growing in Nepal. And if we just proceed ahead for the discussion quickly with the paper, so you guys can check it out. The paper which talks about the abstract, that it's quantitatively and the qualitative analysis of the phytochemical study is get proceed over here. So if we are looking for any kind of phytochemical analysis, so quantitative and the qualitative analysis is really important with any kind of plants which you are targeting. And in that context, they have discussed about like the different assays which is used in the in vitro techniques, with agar well diffusion methods, and accordingly they have used certain kind of uh, targets where the pneumonia, okay, um, the Staphylococcus, Bacillus subtilis, and the Escherichia coli. So this kind of things they have even looked for the extractions, and then they have proceed with the plant to find a richer, what you can say as the targets which they are looking for. So these are their ultimate way. They have looked the flavonoid, okay, accordingly as a significant antioxidant and the antimicrobial or else the bacterial activity. So antibacterial and antioxidant activities get carried out using the phytochemical study. So majorly that has been discussed in the abstract part. And if you just see, guys, majorly they have discussed all the introduction relevant to that. And if them the methods, if you guys can see, that they have proceed by showcasing the plant references. And this is important, guys, that the in the phytochemical they look towards the quantitative analysis, okay, and they have looked to extract based on the plant, means the samples which is get extracted is using the plant data. And then they have proceed with the quantitative screening using the alkaloids, okay, phenols, uh, flavonoids, okay. So these are the targets which they have used accordingly. And in we have certain databases in bioinformatics platform where this kind of screening can be done directly by utilizing the computational approaches like a screening, right? So if you want to like quickly proceed to get to know how the different concentrations and even the targets are available, so we have straightforward databases available accordingly. So what you guys can do is that you can take help of those database to do a quick way of like predictions, right? So ultimately, why to take help of bioinformatics research enhancement kind of approaches? Because it will help you to perform your research accurate and even with a quick way of predictions. But if the predictions we are moving for the quick way, but bioinformatics will provide you the accuracy because the data is properly get mined, curated, and it gets stored in the form of the databases. So that is exactly what you guys have to do in the current era. That definitely you can proceed with like the experimental techniques, but make sure that very smartly you utilize this kind of approaches for the validation, for the screening, right? Where you have a very accurate kind of predictions can be done. Okay, and then you can take help of like those kind of bioinformatics tools where it will help you to design a quick pipelines with accurate way, and if you're proceeding for the validation or the screening, it will give you very accurate predictions accordingly, and that will lead you towards the novel findings. So accordingly they have explained about like what kind of antibacterial activity they have performed using the different bacteria of their target, like Bacillus subtilis, okay, gram-negative bacteria, Staphylococcus, right, Escherichia coli, accordingly. And then followed with that, they have even studied the antifungal activity. Okay, in that context, they have even used the diffusion methods to study their work. Okay, and with respect to the antioxidant, where they have used the proper statistical way to interpret based on the different concentration of the antioxidant study, and followed with that the statistical interpretations of using ANOVA method, right, and using the standard p values they have predicted for the same. Some of the softwares they have discussed, but accordingly that is a statistical way. And in that context you will get to know guys like which are the traditional plants or the medicines which is used to screen the data, followed with the quantitative phytochemical study. And that's why you can see guys they have used with respect to the different concentrations of plant extract, as they have studied the antioxidant kind of study, right? So this is a very complex process. You guys can see that preparing the preparation, okay, screening using the diffusions methods, right, studying the toxicity based on the different concentrations and the mortality rate to get the curve. And then finally in the conclusion you guys can see that they have straightforward mentioned that this present study supports, okay, which particular target they have used the plant and the leaf extract bark. Okay, it's a traditional medicine. Okay, and that is exactly what I'm trying to showcase in this particular reference demo or the tutorial for all of you guys, that if you are using a traditional medicine kind of methods and even the approaches, so how the proper work of antioxidant, antimicrobial, toxicity, okay, whatever you guys are trying to proceed, so how you can lead ahead your further validation very accurately based on like the isolation, purification, characterizations, whatever you have done in the in vitro technique, so that you can proceed with respect to the computational approaches. And that's why it's exactly what you guys have to think about in this way, that how I can utilize the current technology to integrate with this kind of stuff. So for that particular purpose we have the database for us. Basically this database talks about the phytochemical study. Okay, so you can see that it's a PDTDB database which is available to carry out your phytochemical study accordingly, and it will give you a free access of the three-dimensional structure of the plant metabolites, drug targets, and protein and ligand complex. And you guys can observe that it's all about like the plant phytochemical medicinal use, disease targets, symptoms, side effects, drug action, natural remedies, and even the molecular interactions. So ultimately all the things you guys are able to get in this graph, right, in this particular work. And why not to utilize this kind of research in the form of like using this kind of servers. Okay, you can see that exactly what I've mentioned, the prediction servers, okay, the accuracy where you will get with the bulk amount of data. And in that context you can utilize your screening by even having a proper interactive visualization of the 3D structures. Okay, you can download the structures of all the molecules of your interest, and you can record the plants phytochemical disease drug targets accordingly as per your research context. And you guys can see that all the databases are interlinked with this particular server, and that is what you can see, that you can even refer this paper for the reference. I hope that you guys are getting my point, like how you exactly you can utilize your work. And in that context they have provided you the database which you guys can search and browse your related entry. And here you guys can check it out. They have provided a step-by-step tutorial for all of you that searching with respect to the targets you can do, and then you can proceed with the drug name, 2D structure, chemical structures accordingly. You can even browse with respect to the data types, and you can even download to get your 3D structures about your target. And that is exactly what you guys can see, that they have provided a protein physiochemical properties you can get from here. You can get the 2D structures of your targets, whereas you can even draw the targets according to your interest, right? And followed with that, you can even get to know the overall informations about the targets with respect to its molecular docking stuff, targets which has been used, which plant extract they have used, what kind of docking study they got based on the ligand targets. And you can browse based on the different plant targets of your interest. So you will get to know the different plant IDs, right? Extracts based on the medicinal plant, traditional plants accordingly. You will even screen the filters based on the different data types of your interest, right? And you can even use the 3D view to generate your targets for the downloading options, and even playing around the data where you can pull out the 3D structures in the interactive way. So I'm going to show you one example quickly so that you will guys get to know an overview that how exactly you guys can utilize this stuff for the proper analysis and interpretations. I hope that you guys are getting the point, right? So as you all can see that guys, in the browse options you will get the list of ID which we have seen right now, like the same way, right? And then you can click on that ID, and then followed with that you will get the detail of that particular targets. You will get to know the phytochemical IDs, metabolites, targets, plant extracts which has been used with the references for the same. If you even switch with respect to the targets, you can see this, the structures will be get available where you can get into the 3D view, right? Accordingly, and you can get to know very prescribely about your target. And then you can even download the 2D structures where you will get the related plant informations, where you can screen your study and you can look for the proper data analysis. The search options are even provided where you can search based on the target name, the details you can give accordingly to the disease type as we have discussed, right? So disease type can be given, right? You can proceed with respect to the search option. So as you can see that guys, the in the search option they have provided, you can search based on the target name such as the disease name, right? Dengue, or the plant details like a papaya, or the ligand details you can provide with the docking study also. In the structure search, they have given an option that you can search based on the smiles format of the drug, or else they have provided an option where you can make the structure representations of the target. Okay, you can make the benzene ring accordingly, um, and then you can even make the targets with, if you want to make the targets, right? If you want to create certain kind of 2D structures, right? You can add the carbon. Okay, you can carbon is already available here. You can just look towards the targets of your interest to generate. So if you're looking in that context, they have even provided the ID, or the unique IDs can be given. If you even look, like they have given an option like breast cancer. So you can see that the targets and the different plant targets, whatever they have used, they have provided that option. You can search based on that to get to know the 2D structure information, the 3D interactions of the targets. So it is, I think it's very well they have organized, and the search options, the browse options, download options, they have even provided where we can download the structures with the data in the form of the CSV files. The structures can be downloaded in the form of the uh, 3D structures, right? The ligands and even the sequences. So enter the sequences and then you guys can proceed. Some of the examples they have even provided. So it's the best to utilize and just explore your work for the study. So you guys can see that the downloading of the data will be done, and then you can proceed for your further study. So I think comprehensively we can explore the data accordingly to get a quick way about like the structure interpretations, right? And then we can even proceed with some of the tutorials and even the browse options where we will get the exact information about the targets. So I think this database will give you an overview about the target, which talks about like um, the data, right? Updates are still there, right? You can see that PDTD, that is a phytochemical and drug target database. So I just highly recommend you guys that you can try this work to enhance your research by using the bioinformatics platform, where you have a clear-cut overview about the structure, data, interactions, and even the targets accordingly. So in the one shot, you can provide a clear-cut overview that how you guys can proceed for the research point of view, and then you can upgrade your research by enhancement of certain kind of tools with the correct techniques. So I hope that you all of you get the point. Okay, if you want to visit the research paper of this separate research database which we saw right now, so definitely you can just explore with the link accordingly. So I think this is a good overview what you can take as a note and do a practice to explore your study by enhancing your research with the current technology, and try to just think about an objective of publishing your work. So with that note, I think all of you are clear. So I just really all the best to all of you. So I will just conclude. Thank you very much.